Hey everybody, we're going to start in on the last unit of the first semester here, which is metabolism. It's going to involve energy and enzymes, and then respiration and photosynthesis. Okay, so let me move this over a little bit. I think it'll get out of the way better over there. So, um, for life is built on chemical reactions, right? We talk about a lot of chemistry in biology because there's so many chemical things that go on inside our bodies. And so, for this, one of those is transforming energy from one form to another. And the sun is going to give off the energy for our planet, and the plants are going to trap that. Those autotrophs are going to trap that. Those producers, and the solar energy is going to get converted into some organic molecules and some ATP, and then herbivores are going to come along and eat those, and then they are going to pass on some of those organic molecules to carnivores. And you guys kind of understand how food chains work, I'm sure, but they're really after the organic molecules, okay, because that's where the energy is stored. And so if you look at the word metabolism, there's a couple different definitions. The sum total of all chemical reactions that occur within an organism is one way to use that word, or it may refer to like the breakdown or synthesis of organic molecules. A lot of times we refer to the breakdown of molecules being metabolized, right? You metabolize sugar. Um, again, a couple reminders. Organic molecules are building blocks of the cell. The chemical bonds within the organic molecule stores the energy for the cell, right? That's always where the energy is. It's in the bonds. And so how do cells utilize this energy? This is what we're really going to be focusing on, is how is this energy used within the cell? So energy 101, so NRG is just an abbreviation for the word energy. It's just shorthand I picked up in college and never quit using. 101 is like your basic, right? That's your most basic college course is 101. This is just some energy review for you. So energy is just the ability to promote change or to do work by the cell in this case. And so kinetic energy is always associated with motion. Potential energy is always associated with position. And chemical energy is a type of potential energy, and the energy is contained again within the bonds of these molecules. So the first law of thermodynamics, or the law of conservation of energy, LOCE, energy cannot be created or destroyed. You guys have heard that a million times probably. Don't forget the part in red, that it can't be transformed. It can be converted. Okay, and when you convert it, you can never convert like 100%, right? You always lose some amount of energy, um, and that unusable energy is entropy, right? And so we have this Gibbs free energy term down here. And it's a measurement, it's a formula, measurement of the amount of useful energy that a system like a cell can use to do work. So the change in free energy, what we call Gibbs free energy, um, is going to be equal to the change in usable energy, or enthalpy, minus the temperature times the change in unusable energy, or entropy. So it's a pretty basic equation, actually. There's just a few things that you plug and chug, and the math is not hard. I rarely see the AP folks ask a Gibbs question using the math. It's mainly the concept. It's mainly, do you think it will go up? Do you think it'll go down based on certain conditions? So here are some examples for you. So let me make sure I grab the right thing over here and grab the pen. So if we look at this one, and we're looking at an exergonic reaction, right? Energy is released. So here are the reactants. So the left side of an equation, right? So this is how much free energy there is. And then the reaction runs right here. And you see that there is some energy given off. And then you have your products over here. And there's far less uh, energy in the products than there was in the reactants, right? And so energy was released. And so if you look down here, the delta G is going to be negative, okay? Because you gave off energy, okay? Energy was released, right? This is a catabolic reaction. You're breaking it down. And so energy gets released. You're breaking bonds. And so that's going to be a negative delta G, okay? And so these will happen spontaneously once they are initiated. And basically, matter is converted from higher energy arrangements to lower energy arrangements, okay? So let me show you the next example. and it's just the opposite. So you have the reactants here now, and then energy is being put into the system, and then your product's energy is way up here, right? So in this case, we're taking something that's very simple, the reactants are very simple, and we're making more complex products. So in this case, it's an endergonic reaction. We're putting energy into the system, okay? So it's non-spontaneous, okay? So converting from lower energy arrangements to higher energy arrangements, and then your free energy now is positive, right? So we put energy in, so the delta G is greater than 0, and so we get a positive delta G in this case. So if I had to tell you, if I click back and forth here, and one of these is respiration, and one of these is photosynthesis, which one will it be? What happens in respiration and what happens in photosynthesis? Which one is breaking things down and giving off energy, and which one is putting energy into a system and building molecules up? Well, pause and think about it for a second if you want, because I am just going to tell you. Um, for respiration, cellular respiration, if you remember, that's how we generate our ATP, right? And we get it from food, and we break down our food, and we get the energy out of the bonds. Okay, so all that energy gets released, and we take something that might be a really big carbohydrate, and we keep breaking it down, breaking it down, breaking it down until we get to just glucose, and we release all of that energy. So that would be cellular respiration function because it's catabolic, right? For photosynthesis, plants take in the sunlight and they run all these reactions and they're putting that energy in from the sun, and those are sugar building. They're going to use that energy to build sugars, right? So you're going to build organic molecules. So you're going to build much bigger products than what you started with the reactants. So this one will be more for photosynthesis because it's anabolic. So organisms are endergonic systems. We need energy to be put in, right? What do we need for energy? Why do we need it? So synthesis, building molecules, reproduction, movement, active transport, temperature regulation, all sorts of things. So if you stop eating food, if you stop putting energy into your body, you're not going to make it, right? You have to have some food because that's where we're getting our organic molecules from our food so that we can then break down those bonds and use the energy. So a couple more things. Um, Biological systems use exergonic reactions, okay, exergonic reactions to provide the free energy necessary to do endergonic reactions. So this is what's called coupling, when you take, you break something down in order to build other things. And so you can follow all of this over here. It's just talking about the Gibbs free energy over here. Okay, so higher free energy, greater capacity to do work. And then, so the free energy of the system decreases, the system becomes more stable, and the released free energy can be harnessed to do work then. And so, once you finally release all of that, then there's going to be less free energy and less work capacity once it's all been released. So this may help make it a little more sense for you. So when you look at energy coupling, and we use exergonic or catabolic reactions to fuel the endergonic ones, the anabolic reactions. So if you look at this, these are just basic, uh, nondescript reactions here, but we have something here that's put together, right? And so we're going to put the pen away to click. So we're going to take, I don't know why all these words are kind of out of sync, but when I post the PowerPoint, they should all look appropriate for you. So you're going to take two things, you're going to break them down into the individual ones, and when you do that, you get energy, right? And then you're going to use that energy to potentially build, go back one, sorry, to build something else. And so we used, okay, we use the energy that was here that we got from breaking this bond right here. We take that energy and we put it down here to run a different reaction. We needed to add this and this together to make this. So we used an exergonic reaction up here to then fuel the endergonic reaction down below. Okay, so it's a very simple look at it, but that's what's going on. Second law of thermodynamics. So the transfer of energy from one form to another increases the entropy or disorder of a system. So that sounds a little confusing to you, remember entropy is just chaos, right? It's the disorder. And every time you transfer energy, you don't get to transfer 100%. If, whenever you go out running, you're using energy that you brought in through food to give you energy to go run. But you're giving off a lot of heat, right? Your body heats up and you're giving off a lot of heat. So a lot of that energy became unusable for you and you gave it off as heat. That's entropy. So, um, so I think I've actually showed you this slide before. So remember, energy is transformed, the randomness increases, the disorder increases. And so in order to keep systems ordered, you have to keep putting energy in. If not, the universe and the body and your cell would start looking like that messy room right there, and we wouldn't survive very long without it. So again, second law of thermodynamics. So life is highly ordered. We use the energy to decrease or delay the entropy, right? We constantly put in energy into our body to decrease entropy. All your life, you're going to try to fight entropy. Your cells are trying to fight entropy from taking over. And we keep putting in energy to keep that at bay, right? And, a long, 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 long time from now, hopefully, entropy's going to win out, right? Because everyone eventually dies. And so um, at some point, we won't be able to sustain that anymore. But um, life requires the energy input, and that's gonna be what maintains the order and helps your cells function. So, little note about closed and open systems. So when you um, so in a closed system, okay, you have all of this uh, energy here, right? And it's running through this motor or this fan, and it's powering this light bulb right here. And so you're giving off energy, right? Delta G is less than 0 here. The problem is in a closed system, when the water tank drops to this level, right? Now the level's so level all the way across, the fan won't spin. It can't do this turn. And so now we get no energy and delta G is just equal to 0. There's nothing happening. Okay. In an open system, here's the same diagram down here, except we open the cap and we put a faucet here, right? And so now water's constantly coming in, the fan is constantly turning, or the turbine is constantly turning, the light's constantly on, right? And so now, and if this were to happen, if we were to link them together, as long as this faucet keeps putting in the energy right here, keeps adding water, then this will turn and this will turn and this will turn. And that's what keeps the energy input going. So in an open system, you never reach equilibrium. Okay, as long as there's enough input, you never reach it. And open systems are generally more interesting. Life is an open system, right? We have to constantly input energy, and that's why we have all the changes and stuff that goes on. Closed systems are pretty predictable, pretty stable, and pretty boring. So fueling the body's economy, right? So you eat high energy organic molecules, you want carbs and lipids and proteins. You're breaking them down, you're digesting them. That's a catabolic reaction. We capture that energy so we can use it. And so we need a way to pass this energy around the cell. And it wouldn't be very easy to do without this molecule, right? We need ATP. ATP is like your cell's money, that's your currency. And so it doesn't stay around very long. ATP is very short-term energy storage. Okay, so if we want to store that, then we'd have to convert that ATP back into bigger molecules. Like, you remember how we store our energy? You remember how animals store our energy? Could be a good final exam question. Remember, we take the glucose and we turn that into glycogen, right? Long chains of glucose, and that's how we store it. And a lot of then it stores the energy in the bonds until we need it, and then we can get when we break the bonds, then we can start generating some ATP. So ATP, adenosine triphosphate, most of you probably are aware of what it stands for. It's a modified nucleotide, so it's like a nucleic acid. An adenine, and you have, it starts with an adenine, which is the base, then you have the sugar and the phosphate. And you can have AMP, which is monophosphate, ADP, which is diphosphate, and then obviously ATP, which is triphosphate. Okay, so Adding phosphates is endergonic. The more phosphates you put on there, the more energy that gets built into those bonds. And it's really the bonds between the first and the second phosphate and the second and the third. Those two bonds right there are really where there's a lot of energy packed in there. And that's what, when we break those open, that's what we tend to use for energy. Okay. So how does it really store the energy? So each negative PO4 is more difficult to add. If notice all the charges on those phosphate groups there, they're all negative, right? So do they want to be by each other? Nope. Um, but we have an enzyme that sticks them together. And so there's a lot of stored energy in there. And it's most of the energy, a lot of it's stored in that third one because it really doesn't want to be there. Um, it's the hardest one to keep bonded, right? Because you don't want to stick something, a negative molecule doesn't want to be next to two other negative molecules, right? They're kind of like the fighting brothers where they don't want to be near each other. Um, and so uh, it's pretty unstable. And you can think of it like a spring. That third one like gets shoved on there, but as soon as it can, it'll pop off. Okay. As soon as something comes around that it would prefer to be with, it will pop off of there, and it will get put onto something else. Um, and so we'll talk about where that goes and where that happens in a little bit. Um, and so it's constantly, donating that third phosphate, and then we have other enzymes that are going to constantly be rebuilding it. Okay, so ATP to ADP, so you release energy. The delta G changes about negative 7.3 kilocal per mole. Okay, so that's kind of how much you're going to get when you break that bond. It fuels other reactions, and here's phosphorylation again. So remember, phosphorylation is adding a phosphate, right? And so a lot of phosphorylation comes from taking an ATP and breaking off that third one and uh, changing it into an ADP. And then that third phosphate now that's free is going to get added to something else. Remember, phosphorylation is what triggers a lot of reactions to happen. We talked about that in the cell communication stuff last year. Destabilizes other molecules, and an enzyme that phosphorylates is a kinase. So kinases a lot of times grab those phosphates and put them on there, and they phosphorylate molecules to make things happen. There's our 7.3. So an example of phosphorylation here. So you have some synthesis on the top line where it's getting built. You take the two things. This is going to be a little tricky to use my pen and the clicker because it doesn't like me to let me click when I have the pen out. But you're going to add these two things together, right? Here and here. You're going to add them together. And so then how do we add things together? What's that called? Dehydration synthesis. Here's our water molecule, right? So we added things together in dehydration synthesis. So then you're taking here, you have this molecule, you broke that bond, so you have some ATP. A kinase jumps in and it's going to phosphorylate this. So again, back to my pen, this gets added, right? So that's phosphorylation done by the kinase right here. And now we have ADP instead of ATP. Okay. Whoops. Thought there would be another one. And so now, man, I'm not having very good luck with this because the things keep getting in the way. Let's see if it lets, there it goes. So now you have this phosphorylated molecule that is going to go cause another reaction, and now they're going to build that reaction. They're going to put those two things together, and the phosphate will come back off. Okay, and so this will get more clear as we go through here. So an example of phosphorylation, or the first steps of respiration. I know we haven't gotten there yet. We'll do that here soon this week. But in glycolysis, which is the first step, you take a glucose and you start breaking it down. So you can see the glucose is a 6-carbon sugar, right? The green, the 6 green C's there for glucose. And when those 2 red phosphates get added, okay, we have to spend 2 ATP over here. Let me go ahead and click these up and then I'll draw on it. So we have this glucose molecule right here, and here we're going to take ATP, and we're actually going to put energy into the system first. Even though respiration is supposed to generate ATP for us, we have to get the ball rolling. And I'll talk about this with respiration down the road. But for now, we take a couple of ATPs and we break them down into ADPs, and that gives us two free phosphates, right? Because we release those phosphates. And these phosphates are going to be what goes on to destabilize this glucose, and it's going to break them down into two 3-carbon sugars. Down here, you see the 3-carbon sugars on each side. And so that's the first step in trying to break down glucose. We have to put energy in order to start breaking it down. And it really, the phosphorylation is what starts breaking down that glucose. So kind of a little review here of some terms. When you're forming bonds between molecules, right? When you're building things, that's dehydration synthesis. It's a synthesis reaction. It's an anabolic reaction because you're building. And when you're breaking bonds between molecules, that's hydrolysis, and you're digesting, and those are catabolic reactions. So just groups of words that all go together. And that's really review from Unit 1. As is this, dehydration synthesis, right? Go back. Dehydration synthesis, you can see that there's going to be an enzyme that's going to push those two things together, and we're going to create the water, right? We're going to release the water there. And then hydrolysis, we're going to split the water in order to take the sucrose, in this case, and split it back into glucose and fructose. Okay, so hopefully you remember those types of reactions from the first unit. What drives reactions? Why don't reactions just happen spontaneously? Because most of the covalent bonds are super stable, right? So you have to put in some activation energy. You have to kind of get this thing going. So here, here's your reactant right here, right? And so we have to put some energy into the system and get it all the way up here before that thing will start breaking down. And then once energy is released, we end up at an energy position lower than where we started, right? So energy is released in this case. And so we sometimes have to put energy into that to make it go. Okay. Anyway, we just kind of talked about that. So here's your activation energy, very similar diagram on the right-hand side. Breaking down large molecules requires initial input of energy. You have to get it going. They don't just fall apart. Okay, so you have to put some energy in, and you can see the activation energy in the hump going up to the transition state, and then it's going to start breaking down and releasing that energy as it comes down to give you those products. Okay. Catalysts can reduce activation energy, right? Catalysts in biology are generally enzymes. And so if we look at this one here, here's your reactants again. Here's your products. You can already see them down here. We're going to get to the same place. It's just rather than having to go all the way up here and put in this much activation energy, okay, we don't have to put in that much if we have a catalyst. Catalyst makes that happen faster, right? This is why reactions that are catalyzed happen faster, because they don't require as much activation energy. So it decreases the time for that to happen. And they're also reusable, and they don't really participate in the reaction. So the reactants are the same, the products are the same, they're reusable, so we can use them again in another reaction, and they don't participate, so you don't change the product. Nothing actually changes other than it just happens faster. Okay, it's kind of like adding lighter fluid to a fire, right? You can take a match and you can try and light some sticks or some wood, and you can eventually get it to light, okay? And eventually you get that wood to light and you release all of the energy from there. But if you put a little lighter fluid on there and you throw the match on there, it happens quicker. Did you get more energy out of that wood? No, you didn't. You got the same amount, you just got it faster. And here is the rest of the graphics there, which is pretty much what I just drew on there. All right, getting into enzymes. These are our biological catalysts, right? Proteins and RNA, okay, this is going to be what makes them up. facilitate they're going to facilitate chemical reactions. They're going to increase the rate without being consumed, right? They don't participate in the reaction. They lower the amount of activation energy. That's how it happens faster. They lower the amount of activation energy. They don't change the free energy. You get the same energy in or out, okay? Nothing changes there. And most biological reactions require enzymes, highly specific, thousands, hundreds of thousands of different enzymes and cells to get all of these different reactions done that we need done. Okay. So little reminders on enzymes as well. Let's put all this up here. I really wish the words would have turned out better than that. I think they're fine on the PowerPoint. I'm not sure why they're doing this on the video, but I'll double check it before I post it for you. The substrate. The substrate is always what the enzyme is working on. Okay, substrate's a general term. Okay, it's just a general term for whatever an enzyme is working on, and enzymes are going to bind with the substrate and start to work on it. Okay, temporary association between those two. Product is the end result. Active site is where it's going to happen. The enzyme's catalytic site, meaning where that little fit is going to happen between the enzyme and the substrate. So if you take a peek down here, the enzyme is your purple guy right here. Here is the active site, okay? And it's usually the groove that only a particular molecule will fit in. That's why we have all these different enzymes for all the different reactions, because they have to fit like puzzle pieces, okay? And then this is going to be where the reaction takes place, and we're going to cleave that bond right there, and then we have our products, right? We have the end result. So pretty straightforward there. So each enzyme's worked with a specific substrate, has to be the chemical fit, okay? They're not consumed, okay? Which means they can, enzymes can work on tons of reactions very fast. They can keep working as fast as they're running into those substrates. They can keep working on them. Happens super fast. And they can be affected by a lot of different cellular conditions. They can happen faster or slower. Could be based on temperature, could be pH, could be salinity, could be ion concentration. There could be lots of things that affect whether the enzyme is going to work faster or slower. For naming conventions, we've talked a lot about this year, things being ACEs, right? Anything that's an ACE is an enzyme, and generally they're named for what they break down. So sucrase breaks down sucrose, proteases break down proteins, lipases break down lipids. So you can kind of see what's going on. And there are others, there are exceptions. Pepsin, it's not an ACE, but it's still an enzyme. And it's named that way because it breaks down polypeptides. And then you see DNA polymerase is the enzyme that's adding the nucleotides to a strand. So it's not really one that breaks things down, it does a different job. Okay. So an induced fit model is kind of a more accurate model of enzyme action. So when the substrate binds, a lot of times it has to really like click in, it has to like really attach. And so you can see here, again, let me grab my pen. You can see here, here's the active site, right? Here's the active site or the opening on the enzyme, and the substrate here is going to fit in. But if you look at this part of the enzyme and this part of the enzyme, and we look back here, they were kind of out here hanging out. Okay, they were kind of open like this, and once that substrate came in, then the enzyme kind of went through a conformational change, and it clamped down on it, and it kind of squeezed it and locked it in there, and that brings the chemical groups into position to do their job, to do the reaction. Cofactors, okay, just kind of a vocab term. Sometimes, not all the time, sometimes enzymes require other compounds. This is why you need vitamins, minerals, and stuff for specific reactions. So magnesium, have to have magnesium for, oh, let me go back. Just trying to get that thing off of there. Gotta not bring my cursor too low, apparently. The magnesium, that green dot on the left-hand picture there, you have to have magnesium for Rubisco to work. If you don't remember what Rubisco is, it's probably or literally the most important enzyme on the planet. So we will not talk about that until we get to photosynthesis, if that's a clue for you. And then on the other one in here, there's that yellow arrow at the tip of that yellow arrow. There's a really tiny green dot in there, and that's manganese. Okay, that's manganese, another periodic table element that's in there, and that has to be in there for topoisomerase to work. You guys probably haven't heard of topoisomerase, but you will hear about that when we get to genetics. Okay, it has a lot to do with DNA and how it unwinds. So we'll talk about that later. Factors that affect enzymes. Okay, now we're going to go a little quicker here, and there's just some charts and graphs that I'm going to show you about how things get affected. Okay, so we're going to kind of click through these. Enzyme concentration, substrate concentration, temperature, pH, salinity, activation, and inhibitors. We're going to click through a bunch of examples now. So if you look, it says what's happening here. So in class, I would be having a discussion right here with what's going on. So as enzyme concentration increases, the reaction rate increases, right? So if we look at what's going on there, so the enzyme concentrate, the more enzyme there is, the more reaction there is, right? The faster it'll happen to a point, right? Because it does level off. Well, why does it level off? What's limiting? Why can't it just keep going up in a linear fashion, right? Why doesn't that happen? Well, because eventually you have so much enzyme in there that there's not enough substrate. Not all enzyme molecules can find a substrate, so then it doesn't work as fast. It works so fast at the beginning that it tore through all the substrates, and now there's not enough to really keep the speed of the reaction up. So it does go really quickly, and then it levels off. Substrate concentration, kind of the same idea, right? The more substrate you have, the faster it will go to a point. Okay, because if you have a ton of substrate, it will happen faster, and the enzymes will work faster. They'll collide more frequently, but then eventually it will level off because all the enzymes are saturated. All the enzyme active sites are taken. Okay, and there's still extra substrate there. And so in this case, the limiting factor is going to be the enzyme. There's not enough enzyme there to keep up with the substrate. For temperature, a lot of them work really well. A lot of the enzymes work really well at 37 degrees Celsius. I wonder why that is. That's the optimum temperature, right? The greatest number of molecular collisions. Most human enzymes work between 35 and 40 degrees Celsius. That's because our body temp is about 37 degrees Celsius. So most of our enzymes are made to work within that range of temperature. Okay, so if you heat things beyond the optimum, then it's going to disrupt those molecules. It's going to disrupt the bonds. And so it's not going to work as well. And when you heat things up, you can denature them. They can become kind of unraveled to a point where they can't go back. One of my examples, when you heat things, so eggs have a lot of protein, right? So if you make scrambled eggs on your oven, and you and you go ahead and you cook them up, can you unscramble the egg? Can you put it back into the form that it was? No, because it's all made of protein in there, and you just denatured the protein. You put them all into shapes where they lost their shape and their structure, and they can't go back. Okay, if it's cold, if you decrease the temperature, the molecules move way slower. They don't run into each other as much, and so the decreased collisions mean you're not going to have things working at optimum speed either. So, but can you reverse that one? Is that changing the bonds? It's not. It's not changing the shape. It's just slowing things down. So if you heat them up, those should be able to be recovered. Okay, it's just not, once an enzyme gets denatured, you can't recover that. Okay, so this just kind of shows you a few different enzymes here. So human enzymes are obviously working better at 37. Other ones might work better over here, like in geysers where it's much hotter, and you have bacteria and things over there. They have different enzymes, right? And their optimal is different than ours. So there's different enzymes on the planet that work at different temperatures for different organisms. So how do ectotherms do it? So a bobcat, a mammal, that's an endotherm, right? And you can see his temperature line, his black line is staying very constant across there. And so his enzymes are made to work within that range. But look at the temperature range for a snake, right? Their body temperature gets really cold and really warm. And so they have to have enzymes that can work at all different temperatures, or they have multiple enzymes that do different things that might work in different ranges of temperatures. Okay, so really different for them. For pH, kind of the same thing. There's some optimal pHs where a lot of things work, but then there's some other examples. So pepsin, if that's down there less than 3 pH, that's really acidic. Well, where do you think pepsin works? In the stomach. Okay. So trypsin's going to be a little bit more basic, right? It's going to work um, in a little bit more of a basic environment, and a lot of them are just kind of neutral where the red line is for our bodies. Okay, so when you change pH, remember this is why this matters. When you change the pH, that adds or removes hydrogen. That's going to disrupt the bonds. It's going to cause a conformational change, right? So that means when you add those positive or take away those positive hydrogens, those protons, the attractions between the charged amino acids are going to change. Remember in the first unit when we talked about why proteins fold up is because those positive and negative amino acids either like each other or they don't, and that's how they get their shape when they fold. So if we change the pH, that's going to change how they all fold and what the shape will be. Okay, so you can see down here below, the optimals listed out there: the pepsin in the stomach and the trypsin in the small intestines, closer to an 8. Salinity, same idea, salt concentration, there's an optimal for that. And again, salinity has to do with changing the amount of positive or negative ions that are going to be in and around those proteins, disrupts the structure, causes it to fold, possibly denature. So this is why things, are affected by salinity, is because it's changing those bond shapes and then the overall shape. And thus, remember, if you don't have the shape, things aren't going to fit in. The substrates aren't going to fit. They're not going to work as well. So again, they're very intolerant of extreme salinity. The Dead Sea's dead for a reason. It's very, very salty piece of water there. So not a lot of things are going to be able to survive there. All right. So compounds which regulate enzymes. So we have inhibitors, right? Molecules that reduce enzyme activity. Okay. So we have competitive inhibitors, we have non-competitive inhibitors, we have irreversible, and we have some feedback. So we're going to go through types of inhibitors here. So a competitive inhibitor means it's going to compete for the active site. Okay, so sometimes something will get in that active site and kind of be the pebble in the shoe, where if there's a little rock in your shoe, your foot doesn't completely fit in there, right? And so not only is that annoying, but you probably can't function to your optimal level, or maybe not function at all. If it's a big enough stone in there, you can't even get your foot in there. And so this is kind of how it works with this. Point this out over here. Okay, this right here is the competitive inhibitor. It's not doing anything. It just happened to be able to fit in there, and it's blocking the substrate. And so now that reaction can't happen. So that's a competitive inhibitor because it's blocking the active site. So penicillin does this. It blocks enzyme bacteria used to build cell walls. So when you have a sore throat and you have strep throat and the doctor gives you penicillin or amoxicillin, what that medicine does is it goes in, it pokes holes in the cell walls of the bacteria that are causing that, and then it really causes a hypotonic environment. Water's going to go in, the cell wall can no longer hold it in, it's compromised, and so it's going to explode those bacteria. It's going to burst the cells by creating a hypotonic environment. Okay, this one's kind of interesting. Disulfiram, anti-abuse medicine, and so it treats chronic alcoholism. So in this case, it blocks the enzyme that breaks down alcohol. So if somebody's an alcoholic and they really need some help and they're in treatment, sometimes they give them this medicine here, and it blocks the enzyme that breaks down alcohol. Because why would you do that? Why would you want to block that enzyme that breaks it down? So if you can't break down the alcohol, you're going to feel terrible. So if they're on that medicine, if they start drinking alcohol, they're going to start having vomiting and severe hangover factors, headaches, crazy headaches, 5 to 10 minutes after drinking. So if they're on that medicine, they know they shouldn't drink, or they're going to start feeling incredibly bad. Okay? So you can overcome the competitive inhibitor by increasing the substrate concentration, right? So if you saturate something with the substrate, then it can outcompete the inhibitor for the active site. Okay. non-competitive inhibitor. In this case, there's going to be something that's going to bind somewhere else on the enzyme. Down here, here's this thing now, and it's binding somewhere else to that enzyme, and it's causing it to change shape. So now this isn't the same opening that it used to be, and now the substrate can't fit in there, right? And then it can't do its job. So while the active site wasn't blocked, this inhibitor down here attached to a different site, and we call that an allosteric site. Okay, an allosteric inhibitor binds to an allosteric site. It's a site other than the active site. Okay, and so it causes a conformational change. The active site's no longer functioning and keeps the enzyme inactive. Okay, some anti-cancer drugs work this way. Which stops DNA synthesis, right? Stops cells from dividing. It stops mitosis. Okay, that's how a lot of chemotherapies work. They stop mitosis. You have cyanide poisoning down here. Irreversible inhibitor of cytochrome C. We're going to talk about that more when we get to cellular respiration. Irreversible. Sometimes something will bind to an active site permanently, and that means you can never get that back. There's also an allosteric. Irreversible inhibitor. If it binds to that allosteric site permanently and changes the shape of the enzyme, you may never be able to do that function. This is how things like nerve gas, sarin gas, insecticides, how a lot of those things work. So there's something called cholinesterase. Cholinesterase inhibitors, where you can't break down the neurotransmitter acetylcholine. And without getting too much into the weeds here, acetylcholine is one of the neurotransmitters where it jumps the synapse. If you think about your nervous system, remember the two neurons get like this, and the gap between them is the synapse, and chemicals have to jump across there. Those are neurotransmitters. So acetylcholine usually jumps that and brings the signal to the next neuron. Cholinesterase is going to block that from happening. And so what happens is you might get something turned on that won't get turned off, or something that will never get turned on. And so the signals get all jacked up, and you could end up paralyzed or worse. So conformational changes here. This is just a kind of a general diagram with inhibitors and activators. So if we take a look at this guy. So in this case, the active site is this big opening right here. Okay, and then the allosteric site is the one in between the purple U's, okay, and the purple horseshoes there. So you might have an activator that fits into an allosteric site, and that keeps every active site open and available. That can happen, right? That would be an activator where it wants things to happen more or quickly, or you could have an inhibitor. You could have something that comes in here to that site and it changes the conformation of all the active sites. So these are no longer horseshoes, and now this is going to block any enzyme activity from happening. Okay. There you go. Conformational changes, allosteric regulation. So metabolic pathways, getting into some pathway stuff here and some feedback. So a lot of chemical reactions are organized in these pathways, and it's, you know, let me click through here. I think I have my pen off, don't I? Thought it was going to show me something else. So a lot of times pathways, you're trying to get, something from molecule A to molecule G, and sometimes there's a lot of enzymes that makes that happen. A lot of times they get divided up into much smaller reactions along the way rather than one big one, so that we have more control. Okay, if you have more enzymes in there, we can, there's intermediate branching points. You can send signals or other reactions different ways. You can stop things before making the end product. So it's really more efficient if you have these branching points along the way where it takes a lot of little steps to make something happen. It actually gives you more control and more regulation for what's going on. So they get organized into groups, and you're going to get very familiar with this picture in a little bit, because that's a cellular respiration picture, right? That is the electron transport chain. And so we'll take a closer look at that down the road. But again, endergonic and exergonic reactions are happening in that picture, where you're taking reactions that are fueling other reactions. Whoops. Just real quick, again, you're going to get this in much more detail when we get to respiration, but this is happening in the mitochondria, right? So feedback, so regulation and coordination, so step-by-step pathways. If you're really looking at feedback here, so A is going to turn into B by enzyme 1. B is going to turn into C by enzyme 2. And you have these stepwise reactions getting all the way down there to G. And if we make a lot of G, if we start making a lot of G, whatever that is, and it starts accumulating, well, it can go back and inhibit enzyme 1 because we don't need any more G. We don't want this pathway to keep going and just making stuff and making stuff and making stuff that we don't need. And so when so much stuff starts getting made, then a lot of times it becomes an allosteric inhibitor of the first reaction so that we can then shut that pathway down until we need it again. So that's feedback inhibition. Another like real life example here. So if we look here, our initial substrate is threonine. That's an amino acid. Okay. And here's enzyme 1, which there's the name for it, threonine deaminase. And so intermediate A, intermediate B, intermediate C, intermediate D, all controlled by these enzymes. And here's our end product of isoleucine, another amino acid. Okay. Well, when we get a lot of isoleucine, when we start getting too much, isoleucine comes back all the way up here. If you read this, it's an isoleucine in an allosteric site. It's going to bind, and then the active site is going to no longer bind threonine, and that's going to shut this pathway down. So it'll build isoleucine to a point, but it won't just keep building it and building it and building it. It'll build enough, and it'll shut itself down. And we are done. Make sure you can. There's some questions. I don't know if you guys ever go back and do these for review, but these are always great to do before quizzes and tests, to see if you, and don't look them up. See if you can really answer them. If you can't, then you got some studying to do. So anyway, that's kind of the purpose of those things. That was a lot. It was really long. Hopefully you broke it up. But if you didn't, that's good. Good for you. There's going to be a lot of material this chapter. Only 3 sets of notes, but each one of them is kind of long, kind of like this. So anyway, I hope that was helpful in understanding energy and enzymes, and we'll talk soon.